Welcome to the Microscopists, a bite-sized bio podcast, hosted by Peter O'Toole, sponsored by Zeiss Microscopy. Today on the Microscopists.
Today on the Microscopists, I'm joined by Joerg Bewersdorf of Yale University, and we discuss his career path from wannabe theoretical physicist to cell biologist and biomedical engineer.
I realized I was actually much more motivated by doing physics, which other people cared about.
Staying away from his lab equipment.
But when I go into the lab and I approach the microscope a little bit too closely, my lab members start to stiffen up and hopefully he's not touching anything.
The very important question of what constitutes dessert.
There's a whole kind of classification of desserts, which I find is very important.
And gingerbread microscopes.
The problem is after a few days, the microscope collapsed. So I think the humidity was too high.
All in this episode of the Microscopists. Hi, welcome to this episode of the Microscopists. I'm Peter O'Toole from the University of York, and today I'm joined by Joerg Bewersdorf from Yale, the School of Medicine. George, hello, how are you today?
I'm great, thank you. How are you?
No, I'm really good. It's been a busy day, but I've been really looking forward to this. So I've got lots of questions. Actually, we're running a course recently, and we actually had a technical problem with one of our demonstration slots on spinning disc, because I believe that the Andor Dragonfly was sent over to your course over with Alison North, and it hasn't arrived back in the UK in time.
Oh, what can I say?
So I presume the Dragonfly was beautiful and worked wonderfully.
You should have seen it. It was fantastic. It really was the best ever. I'm so sad that
we had to invent a whole new practical session where that would slot in. So obviously we got the 980, we got the Airiscans, the Lyras, but we didn't have the spinning disc. So actually we did talk about you, oddly enough, not because you had my spinning disc, but we talked about expansion microscopy, which we'll come to later. I'd like to start off slightly differently today. I'd like to ask you, when you were 10 years old, what did you want to be?
Astronaut.
Astronaut. So very similar to Eric Betzig. So when you were 16 to 18, what did you want to be?
A cosmologist, kind of a physicist, kind of like theoretical physicist, I guess. Yeah.
Okay. So none of this is quite panning out yet with your current career. So today, if you could do anything, what would you be?
I really like where I'm at right now. I think, yeah, I like my profession. I like the field I'm in. Yeah.
Okay. So let's come to that. So obviously you said at 16, 18, you like the physics and the cosmo side of things. And I think your first degree was in physics. Is that correct?
Right. Yeah.
So that was over. Actually, I looked at you at Heidelberg, Freiburg, Glasgow.
Yeah. Freiburg, Glasgow, Heidelberg. Yeah.
That's moving around a fair bit for your degree.
It wasn't that uncommon. So I was still in the at that time we got not a master or a bachelor degree. We got this diploma degree, which was a 5-year degree in Germany. And there was this intermediate exam after 2 years, and I took that in Freiburg. And it was quite common to then do a year abroad after that. And for that, I went with the Erasmus program to Glasgow. And after that, instead of going back to Freiburg, I just decided to go to Heidelberg because the courses they offered, the specialty courses they offered were a little bit more aligned with what I then wanted to do.
So I presume Heidelberg was a very big influence on your career going forward.
Yeah, actually, I think the biggest, there were two important breaks, I would say, in my career path within the physics world. The first one was in in Glasgow, actually, where I was introduced to medical physics. Before, I was always considering myself, I become like this theoretical physicist who works on physicists' problems. So, I don't know how old is the universe, these kind of questions. And then I realized I was actually much more motivated by doing physics, which other people cared about, rather than the people just within your own community. And the medical physics lecture or course I attended in Glasgow was really fascinating to see how you could use physics to really build new MRI scanners or CT scanners, etc. And that really became a big motivator for me. And because of that, I went actually to Heidelberg then. And then I thought I'd become a medical physicist, like, maybe work for Siemens or Philips or so, and build like CT scanners or so. But I thought, I should take one optics course before I finally go into this field, because optics is kind of related and you probably need it. It's super boring. I took, like, what we did in high school was like, I mean, seriously, it was just a lens here, a few lines there, and they all meet. And it was sines and cosines adding up, but like, nothing really fun going on there. But I thought I better take that course because that's my last chance. And that course was taught by a young assistant professor, essentially, who had to kind of more in a adjunct role, who had to fulfill his teaching obligations. And he offered this 2-week block course in Heidelberg over the summer break. And I thought, great, I'll do this for 2 weeks. And then after 2 weeks, I'm done, never optics again. Well, it turned out that that course was actually really interesting. And that assistant professor, whose name you might remember is Stefan Herrl, he really managed to connect these optics concepts to really the latest and greatest in microscopy. And I thought, wow, this is actually really cool. And that was essentially then where I shifted to kind of the, I don't know how you want to call it, biophysics or world. Yeah. So, yeah.
So actually, it's interesting because obviously that was before Stefan got his Nobel Prize.
Yeah.
And I think we forget.
He had just come back from Finland. Yeah.
Yeah. So I think we forget that a lot of so Richard Henderson, Eric Betzig, who are on these podcasts, they're known for their Nobel Prize outside their field, but you maybe forget sometimes just how they've inspired many other very successful and leading and inspiring people outside of their lab that so kick-started their career. It's not just about their ideas that they've developed. They have this legacy that's spreading out.
Well, yeah, I'm not sure I would count myself into that legacy, but I think you're right. Yeah, I think there's something. They have this. I don't know. They have such a high level of enthusiasm, which is really contagious. I think that might be part of the secret there.
So you wouldn't count yourself as enthusiastic then?
Well, that maybe yes. Yeah.
Yeah, absolutely. I've seen your presentations, and they are inspiring. In fact, your last one, it's one of the very few that I actually listened to and thought, okay, I don't know if we have a need, but I'm going to try this. And so actually, I have a student at the moment working on expansion microscopy. I've heard about it before, but it was seeing the results, the very latest results, that I think changed the essential need for us to start to develop this at York for our end users. So we actually have Savas is working on this as an intern student at the moment, doing his PhD and trying to work this up. And I have lots of questions after this podcast.
Okay. Yeah, I'm happy to answer that. I think I'm super excited about that stuff right now. Yeah.
So after your PhD, you did a postdoc at Max Planck. Again, I think you followed with Stefan with that. Is that correct?
Yeah, I already did my. I essentially stayed after that optics course. Okay, I essentially immediately joined Stefan's lab first for an internship, which then turned into a master thesis, which then turned into a PhD thesis, which then turned into a postdoc. And I essentially stayed with him for 8 years or something like that. The PhD was semi-independent from Stefan in that it was actually at Leica. But Stefan was still my PhD mentor during that time.
So there's a train of thought that you should move around, and we'll come to that in a moment. But you did your undergraduate sort of with Stefan, you did your PhD or master's, PhD, postdoc, all with Stefan. So arguably you stayed in the same group. Did anyone say to you that that is career suicide? You should move, even though you're working in one of the premier labs in the world? Did anyone actually suggest to you that maybe you should move somewhere else?
And actually, no, but I think this might actually, I only got introduced to that thought when I moved to the US. Maybe it's not so much part of the German academic culture, at least at that time, or maybe it was just my immediate neighborhood. But yeah, no, it was, I think it was clear that we were up to something great there. And it was just super exciting watching kind of this happen around us. Like when I came to Stefan, I think I might have been the first person who started in Germany when he in his lab when he came. I mean, he brought people, including Germans, with him when he came from Finland. But I was kind of his first new hire in Germany, and we were like 5 people in Stefan's lab, maybe 6 or so. When I left, there were 35 people. Oh, so I mean, wow. It's really just, you could see the whole thing kind of grow and become bigger. Yeah, and I was just smack in the middle of it. That was really fun.
That's a lot. So actually, I'm very similar. I did my research project in a lab of Richard Cherry and moved into single molecule imaging back in the early 90s. And Gaussian fitting, and why leave a really inspiring lab? There comes a time where you have to strike out yourself.
Yes, yeah. So, and you're right. I mean, when I then moved to the US, it was also exciting to suddenly be in this new environment and get all these new inputs. So yeah, I then started to appreciate what I later on learned was good advice to actually once in a while change your environment. I think it does push you to new levels and gives you new insights, and yeah, it makes you develop your own focus maybe a little bit better. Yeah.
So you moved obviously to Glasgow for a short time and then back into Germany, and then you moved over to the US. How, you're very young at that point. How did you cope with those moves? Was it daunting? Was it exciting? Were there problems with moving those physical distances?
I think for me, it was less of a problem than for my family. I mean, I brought my wife and my 2-year-old with me, and my wife was pregnant in, I think, 7th or 8th month. So, and I really have to give a lot of credit to my wife that she did that move with me during that time. And yeah, for me it was not that hard, right? Going from one institute setting to a new institute setting, but the language of science is the same everywhere essentially. But yeah, but moving kind of to a completely new country, learning how the preschool system or the kindergarten system works, how make new friends and all that. I think that was the bigger step there. And we never planned to stay that long, I think I should also say. So it was always intended to just do that for a few years.
That was my next question. How long did you say you were going to be there?
I think we were thinking of going for 3 to 5 years. Yeah. And that's 15 years ago, 16 years ago. Yeah.
So what was the biggest challenge, would you say, when you moved over to the US from Germany?
I'm not sure it was the biggest challenge, but that was certainly something which I still remember very well. And that is the different way of communication, which you're not taught like anywhere in school, but the kind of different level of politeness, or like let me see if I can find a good example. If I would have asked in Germany somebody to collaborate with me, and they were not really convinced, they would say, "Oh, I'm not convinced," or "No, I don't have time for that." They would give you a kind of a straightforward answer, and you would say, "Okay, he doesn't have time for it." If you in the US, what I experienced was more like, oh, that's an interesting idea. Let me think about it. And then you say, oh, they like it. They find it interesting. They think about it. And then you wonder why they never get back to you. So that kind of thing I had to kind of learn how to read these social cues, which were a little bit different.
So it'd be interesting to know how it was perceived when someone approached you and said, "Can I collaborate?" And you say, "Oh, I'm not so convinced by that." How they took it.
Yeah, I think I was cautious enough to not step on too many people's toes. But I've certainly explained the German way of a more direct answer to American colleagues or other colleagues, and told them to not take it personally when they get a kind of a more kind of straightforward, maybe not very diplomatic answer.
So you've lived in 3 countries. So I think I know the answer to this, but I'm going to ask you, what's your preference? Germany or UK?
I don't know. It's probably Germany, because it's in the end we are still you can't really unroot a tree. So I think we are still somehow connected with the culture there. And yeah.
Germany or USA?
It depends on which level. So on the work level, I really like working in the US. I like the environment I'm in. I like kind of the dynamics. I like the challenge that you're permanently facing. Like you need to get your next grant and all that. While it's really stressful, it also pushes you to really continue working and thinking and staying ahead and on top of a topic. So that I think is good for me, and I like that. Like privately or kind of culture, I think both countries have their strengths, but we would probably connect a little bit. I mean, we have great friends in the US and really deep friendships. I really like that. But like from a social environment perspective, I think we feel a bit more comfortable in Germany still.
That was very well put, because you could have either lost your German passport or your green card in America. One of the two. You'd have answered that.
I've actually both now. Since half a year, I have both passports. Yeah, so.
Yeah, they were very well answered on that question. Coming to your research, I wouldn't know where to start, because you have fingers in many pies. Actually, pie is probably a bad use of terminology when 4 pies also associated in there. But you've had interactions with Palm, Storm, Paint, Stead, Pan-X, Pan-XM, if you're microscopy honest. What's your favorite technique?
I'm afraid you'll probably get a similar diplomatic answer there. But I actually believe that. I think all these techniques have their strength and their challenges. So we are, at the moment, we are still working actively in the expansion microscopy research and push that frontier. We're pushing on the Palm and Storm side, and we are also working still on the STED microscopy side, and really try to push these to the next level, or what we think is kind of a missing feature or characteristic they need. And it's really, none of these techniques can fully replace any of the others. I really love about the expansion microscopy that you can do it with any microscope now. You can that it has this correlative aspect now with these Palm stainings that we introduced there, which I think is super important and gets a lot of my colleagues, cell biology colleagues, excited. But do we know how well the structures are preserved at the 10 nanometer scale, or really low? Or let's say you want to measure the diameter very precisely of a nuclear pore complex, or like that particular protein within a nuclear pore complex. At the moment, I would probably say, I'm not sure I trust it to the exact measurements at that really small level. And I would probably go to a Palm Storm microscope to do that. Also, similarly, if I want to count how many proteins are in that nuclear pore complex. And then and the STED microscope I really like when it comes to imaging deeper in the specimen, or you you see what you get, or you get what you see, whatever the right saying is there. But you don't need to worry about like image processing artifacts. You don't need to worry too much about for fixation artifacts there on the stat side. So I think they all really have their strength, and I probably missed a few here, and I missed a few weaknesses here. But the point is, I think they all are important developments, and continue to stay important developments.
And actually, I think Palm, Stormstead, actually quite difficult concepts to describe briefly. But Panex is probably quite easy to explain to a layperson. Of course, the audience, some people may not know what expansion microscopy is. So could you describe it? Really basic concepts of what expansion microscopy does, how it works.
So expansion microscopy, which is a technique where you have a fixed sample. It's always very important to point out the sample is fixed and dead. Okay, and now you embed it in a polymer matrix, and you then let that polymer matrix grow, essentially by soaking it in water. Like when you put a gummy bear in a glass of water, it will swell. Or if you would have one of these little sponge animals that your kids put in the bathtub, as soon as they get wet, they will kind of grow. So that will happen to now this cell. And if you have broken the bonds of the molecules in the sample before that swelling process happens, by, for example, in our case, denaturing the sample, then the molecules of the sample will grow with that gel and stay relatively in the same locations to each other, just that all the distances expand by the expansion factor of that gel. And the magic now is that you can now image your now expanded cell with your normal microscope, and the microscope is still limited to the same resolution limit, but because the sample is so much bigger, you can now see these small details there. And then later on, after you record the image, you just shrink it on the computer back to its original size, essentially adjust the scale bar for it. And now you have, let's say if you have a 20-fold expanded sample, you end up with an image with a 20-fold improved resolution over what you normally could get.
And without any special equipment.
Yes, I mean, that's a little bit sad for me as an optical physicist, but by developing these techniques, I'm essentially making myself obsolete here. But it's true, yeah. We are just using a standard commercial confocal microscope at the moment to record these samples.
So that's interesting because that is very chemical. The whole process is chemistry driven, really. You're a physicist, and you're using this with biological samples.
Yeah.
Are you a chemist, a physicist, or a biologist?
I'm definitely not a chemist, I have to say. That is, I'm always getting very quickly to my limit there. And I rely on my really excellent grad student, Ons and MSAT, to really explain to me the chemistry that's going on there. I'm not sure I would call myself a biologist yet, but I've picked up a lot over the last kind of 10 years or so. And at least I hope I'm not embarrassing myself anymore when I talk to my cell biology colleagues. I would still say I'm a physicist, but actually maybe even more so than a physicist, I'm an engineer. And the difference there, I think, is the attitude. I want to get stuff to work more so than understanding. I'm focusing on the goal rather than the way towards the goal, maybe. And that, I think, is kind of a certain mindset that helps.
So that actually brings me to one of my quick fire questions I was going to ask. Are you a detail person or a big picture person?
I'm a detail person who forces himself to see the big picture. It's a constant push and pull. It's a constant compromise. I need to remind myself all the time when I'm reviewing or revising a manuscript to not focus on fixing every comma here, but actually making sure that the message doesn't get lost. Yeah, so and I think it's important at the same time to, and seeing the big picture is the most important thing, I think. So reminding yourself of that all the time.
So talking of big pictures, you're in your office at the moment. So for those who are listening to the audio podcast, George is in his office with a big whiteboard behind him. And the whiteboard has a few, by looks of it, chemical structures on it.
No, these are supposed to be cellular structures, actually. Yeah.
Well, you didn't say you were a biologist, so I'll forgive you.
And I'm not an artist, obviously.
Well, a good artist never makes a good scientist, and a good scientist never makes a good artist. That's my excuse for being rubbish at drawing. How long have those pictures been on your whiteboard for?
Wait, let's see. When did COVID start? Yeah, it's nearly 2 years there. Yeah, I'm not sure they even come off anymore. You know how this stuff really kind of encrusts itself into the board after a while.
I think there's a tendency, and a lot of people, and the offices I walk into, all the academic offices I walk into, even my own whiteboard, which is now clean, because I realized you go in and people's whiteboards, the contents changes in little patches, but not the big stuff. And there's some stuff that almost never vanishes. So do you have parts of your whiteboard that you're really proud of? You like that, it's part of you, it's something you've got on there that you leave on there?
No, and I don't think that that's actually the argument why you see these patches in other people's labs. I think it's because that stupid pen doesn't come off the board anymore after a while, and you need to really rub really hard, and you give up after like finishing that large area. I think it's more like a pain. You reach the pain point there, and then stop.
You know your optical lens cleaning fluids. Just use that.
I know, but even they sometimes don't work that well. I think it's actually not so much it's a mix of the pen and the underlying material. So I'm actually a big fan of using glass boards now, writing on windows, or we actually have in one of our labs like a glass panel to write on. I think that works much better. That's if I would form another side business, I would go into that.
That's only because you can't clean your whiteboard. You started to write on your windows.
Yeah, well, yeah, exactly. And why not, right? You have a nice view while you actually explain something to someone.
So, back to work, as in more serious work. With all the different technologies, how big is your team?
About 15 or so. I always underestimated. So I think it's probably around 12 to 15. We can go on the website. I think our website is current. Actually, no, it's not. We just have a new lab member starting today. Yeah.
So how do you juggle managing? Because it's not just managing the staff, the team, the postdocs and the PhDs. It's managing the projects that they're also running with, because they'll have disparate projects, especially when you're working on the Palm, the Storm, the Stead, the Panic. How do you actually? It's a lot of your time, because it's lots of different bits of support you're giving. So are you a hands-off kind of supervisor that's there for advice and inspire, or do you actually get in the lab or get into the gritty details with them? How do you manage that time to make sure it's effective?
I try to spend about half an hour to an hour with every lab member individually every 2 weeks. It used to be every week, but I don't find time for that anymore. And we sit down and talk about the data and talk about like where the project goes. We troubleshoot maybe or discuss where the project gets stuck. So that's how I stay involved. I would say it's not really hands off. It is fairly hands on even on detail questions, as we discussed earlier. I'm detail oriented, sometimes too much, right? And then my lab member needs to remind me that that's not why they came to talk to me. That they can figure that part out, which color of a pen they should use or whatever. But what I'm missing a little bit is the hands-on experience in the lab, and it's when I reached that stage. It doesn't sound like you have reached that, but where I'm and I'm envious for you. But when I go into the lab and I approach the microscope a little bit too closely, my lab members start to stiffen up and like, hopefully he's not touching anything, kind of. And yeah, so I learned to not to interfere anymore in the experiments. Yeah.
It may be the opposite. It may be when they run out of ideas of how to solve a problem, they then drag me into the microscope at that point, which is great. And then I'm like, because I can troubleshoot, but the software's changed.
Oh,
and
yeah.
like, okay, can you do this? Try this, try that. So now I'm directing more because some of the software is so alien. It's hard to get to grips. And some of them, the courses that we run enable me to keep up to date with the software. So I find that really useful. But yeah, otherwise, yeah, you do lose touch. It's quite hard to keep in contact with the software changes. But the concepts, microscopes, optics, they change, but they don't change a great deal. If you've got the theory, you can work with it. So thinking of courses, so you have the big course over in the US that you've not long finished doing. So how many delegates on that?
What do you mean by delegates? Students or
students? Participants.
We had students. Well, actually, that's a good point. We were discussing how we should call our course attendance. We felt students is not necessarily the best term for them, but yeah. So we normally have 24 course participants, and this year we had 16 because of kind of COVID restrictions. We wanted to make sure that for the most important practicals that every student had their own microscope stand.
Yeah, and better distance. Very similar here. We decreased by about a third for the same reason. We call them delegates. Attendees is a good answer, or participants. Again, students, and you've got academics attending, and we always worry that they might feel a bit odd being a student or being called a student again.
Exactly.
So they probably shouldn't. They're probably quite proud that they're a student again. Which probably means they're going out drinking every night.
Exactly.
I'm probably too old to be doing that. So you obviously, do you enjoy teaching on the courses?
Oh yeah, I think it's one of the highlights of the year because it gets me. I mean, you know what I'm talking about, right? It's a very intense atmosphere, working very closely with people, I get my hands on instruments I normally don't get them on, meeting tons of people and participating in the enthusiasm and the excitement that the delegates express when they learn these techniques. It's very rewarding, I think. It's a ton of work, but fortunately I do this together with Alison North, and Alison is lifting the heavy load here on the course organization. I'm very happy for that. Yeah,
I could have predicted that part. Alison's very good at organizing, that's for sure, as well as being an excellent microscopist and tutor. But yeah, things won't fail if Alison's involved.
Yeah.
She'll make sure, she'll have thought it all through. She'll have almost run it through in her head in real time to find the faults.
Yeah.
I'm very much not a detail person, big picture person, but Alison does a big picture and that detail, which makes sure, a very good person to work with.
Yes, no, I'm super happy. I hope she's not kicking me out anytime soon.
Where was the course run?
Sorry?
Where was the course run? Where
it was at the MBL in Woods Hole, so okay. Marine Biology Laboratory. It's one of the two courses they usually run every year here in the US. And then there are a few other big courses like in Cold Spring Harbor and Mount Desert Island Marine Biological Laboratory. But I think ours was the only one which actually couldn't happen this year. I think we just got lucky in that break between the last wave and the Delta wave coming. So yeah, I think we in hindsight can count ourselves really lucky.
And so can the participants.
I think so, yeah. They were very happy, it seemed. Yeah.
Can you imagine the number of PhDs, postdocs, technical staff that have now had 18 months of being in position being in a lab, because you can't work from home very well, so you have to be in the lab, but they've not necessarily had the training or the added training to really to make it really work effectively. So I'm sure they're very grateful to have got in there.
I think the training is actually probably one of the aspects which suffered the most in the pandemic. At least that's what I saw here at Yale. And like a senior lab member who already works kind of independently, they know what they have to do. They can work in a more isolated manner. But a new lab member, I think we underestimate how much you pick up by just watching somebody else doing it, or being able to just call across the aisle or so. Do you know where the so-and-so is? Or can you show me briefly how I close the lid on this whatever bottle? Like these things, or somebody looking over your shoulder telling you, what are you doing? You are supposed to put a pipette tip on top of the pipette, like stuff like that is something which was missing essentially for 18 months. And I think,
I think that had been very daunting for new starters into a new environment. But to take you back, what was your first microscope that you used? Can you, not in school, so when you went to a more advanced microscope, what was your first big microscope that you used?
I used a Leica a TCS and T confocal microscope. So that was like the latest Leica confocal that Stefan Hell just got in his lab, and I, as the new lab member, was kind of witnessing having it set up, and I was the first who was allowed to tamper with it. So coupling a 2-photon laser into that microscope. Yeah, I think the Leica service people were pretty scared. Like I think I was a little bit oblivious, which was good, but like having like this kind of I was not even a master's student at that time. This intern essentially taking screws out of this just new, like brand new confocal microscope to like add a mirror or so somewhere in the middle. That was scary.
So thinking of the multiphoton side, this is a really powerful laser at this point. It's beyond your normal confocal lasers. It's a very powerful laser. Did you at any point worry that you'd damaged a photomultiplier tube by blinding it with too much power? And how many biological samples did you blow up trying to get the laser power right?
Well, we definitely blew up I blew up my fair share of biological samples. I didn't destroy photomultipliers to my knowledge, but I haven't compared like an old and a new one side by side. So I don't know. I would not be surprised if one or two suffered a little bit in that project. But yeah, like blowing up samples by focusing accidentally on pigmented areas in the sample. Yep, I've been there.
Have you ever tried to pop popcorn using the multiphoton?
No, but that's a good idea.
Yeah, I've not tried it. I have to try it. I keep thinking about it, and we teach it on our course. I'm gonna find some later and take it into work. A bit of oil immersion, bit of butter and salt. See if we can pop a bit of popcorn with the lights, as long as I don't break the cover slip first.
Yeah. Oh, we managed to burn 2 objective lenses in the last year. Not even with a multiphoton laser, just with a CW laser. And I've never seen anything like that. They have a little crater in the front now. It's not somewhere in the inside. The front lens is not smooth anymore. It now has a little kind of crater in the center. It's crazy. I didn't know that.
Shame the manufacturer of that lens, but I won't.
I am pretty sure it's not the manufacturer's fault, because like it's really not any layers, so it's the front. So I think what happened is the microscope which we built had it was kind of automated platform, and it had it crashed essentially in a long-term, like 24-hour experiment, and it stopped moving the stage, and we hadn't, and the software had a glitch that it didn't turn off the laser. So the laser was constantly focusing onto the same spot of the cover slip. And at I don't know what happened then, somehow it must have heated up so much that I still don't understand how the front lens really caught. I mean, it's glass. I mean, how are you supposed to create a crater on the glass surface?
And you should probably stop there, because you've had the stage stop working. The laser fail-safe for the laser blanking has failed. If your health and safety are listening to this, you're in big trouble tomorrow.
Oops. Well, yeah.
So you get these stressful times when you realize that this has happened. What do you do outside of work to relax?
Spend time with family. I think I like cooking. Preparing kind of good dishes.
So what's your signature dish?
It's probably on the dessert side, or so. That's probably my major passion. I think it's kind of a creme brulee or something like that. Yeah.
A creme brulee.
Yeah.
That's as exciting as it gets.
Oh, I think there's a certain art to it to get it just right.
So, do you go for a creme brulee that's super thin, or do you go for deep with the top?
Yeah, I think it's more like like that.
The deeper
ones,
rather than the really thin ones with a very delicate top.
Yeah.
Have you ever done a deconstructing one where you do the sort of the glacial sugared and put a shard of it into your creme brulee?
No, I guess I'm a traditionalist there.
So
it's just that my wife doesn't like me handling the blowtorch in the house, so that's the biggest problem.
You're an engineer. Oh no, I've heard what you did to lenses. Yeah, maybe best.
Well, maybe we can use a laser for that.
You can make a whole pattern really well with it.
Yeah, like a 3D printer, right? You can write into the sugar coat.
So if you had dessert, would you be an ice cream man or a chocolate man?
So, wow, okay, now you get me started. There's a whole kind of classification of desserts, which I find is very important. And ice cream is a proper dessert, as is a creme brulee, but chocolate itself is not a dessert. That's a sweet, which you're not eating after a meal. And cake is also different. Cake is not a dessert at all. Cake is cake. So that's an argument I always have to have with my American friend.
So I'll argue back. What about a nice chocolate torte? With a crisp pastry bottom, dark chocolate, 70% dark chocolate rich as a torte?
Yeah, but yeah, I won't eat that. That's something you eat in the afternoon with a tea or coffee. That's not a dessert. A dessert is something you eat after a meal. By serving that cake in the evening after your main meal, then you actually cheat yourself of that afternoon meal, because you already had. So you should have both separated. If you put them in two different categories, then you said, I had one dessert today and I had one piece of cake today, rather than two desserts.
You see, you missed the point. There's no reason you can't have it at both times a day. In fact, we taught courses. We always take the delegates out for meals. And the one thing I insist on being on the dessert, we do kind of go to restaurants and we kind of dictate what should be on the menu. Because we kind of know what the delegates will go for, tried and tested, and we've honed into what everyone will ticks many boxes. Chocolate torte has to be on the end menu. And the number of times I've ended up with 2 or 3 chocolate tortes in front of me, wow, you have to oblige. Sleeping that night's quite difficult, especially when they are properly dark chocolates.
Yeah, that's only an experience I had with if you hand out drinking tickets to people. Everyone gets 2 beer tickets, and it actually turns out, like, at conferences and all that, turns out if you keep your eyes open, who is drinking soda, and you then ask, hey, do you have a spare drinking ticket? You can get your fair share, which also makes it difficult to sleep later.
You can do quite well out of it. That's for sure. So thinking of conferences, out of everything that's happened over COVID in the past and everything else, virtual or physical conferences, virtual or physical meetings, what's your preference?
Physical. Virtual doesn't really work for me yet very well. I don't know. I miss the meeting friends and colleagues. For me, often at the conference, it's not so much the actual talks which excite me. It's the hallroom discussions I have, running into somebody or looking at a poster I didn't expect, and like I know it's these chance encounters which I think is, for me, often what I bring back from a conference, where I learn something unexpected. It's usually not the big plenary talks, and it's usually not even the regular talks. It's these side events.
Yeah, no, that's fair enough. And do you enjoy traveling? Because obviously you get invited to lots of talks all across the world. What's your favorite type of conference? Long haul flights, short haul flights, back into Europe, elsewhere?
I don't like flying so much anymore. I like sitting on a plane for a long time. Is not fun, and the time zones, or like time zone change, also bothers me more and more, I have to say. So I don't know. I haven't traveled that much over the last 18 months. So at the moment, it sounds better than it probably is. But that's the only disadvantage of the physical conferences, the travel, right? So being able to just call in is a huge advantage.
So with all the techniques that you developed, you must be writing lots of grants, and the grants are obviously diverse. So you're never ploughing the same field. So you have multiple runners in a race, if you like, which must help your grant success rate. What would you say you also patent some of your work?
Yeah.
How do you balance patents over publishing the work? Or even how do you know there's going to be a patent in this, you've got to write a grant application, but you don't want to give too much away from the patent side?
That's not how I see. I don't see there's a conflict from my perspective. I think you just need to make sure you file the patent early enough. And fortunately, at least in the US, we can file these provisional patent applications, and they can be turned around if necessary. I mean, you're not making friends in the tech transfer office with that, but they can be turned around in, I don't know, 48 hours. So what you just need to keep it in mind, from my perspective, that before you, let's say, put your paper on bioRxiv, that you just a day, or ideally a week or 2 weeks before, you actually make sure you file that provisional patent application, and then you can follow up in the next 12 months to file the proper application. And then we are not holding back then information from the paper for strategic purposes. I think that would be dishonest, and it would also not be in the interest of the funding agencies who funded the research, so and not in the interest of universities. So I think these things actually go along quite well. In a way, the paper is even advertisement for the patent, right? I mean, nobody would know about the patent, or ask you about licensing it if you wouldn't openly advertise for it through the paper.
How helpful are the tech office in putting the patent together? You're a scientist, you're not someone who writes patents. So how much do you get? A lot of support at Yale for that?
The way Yale does this is they put us in touch then with outside faculty, outside patent attorneys, and they help us raise these patents. And there's always you need to bridge kind of. They don't have the knowledge on the field, and that's always kind of difficult to explain to them. Then, and you certainly, it's going back and forth, but they can certainly help you get this into the right claim language, which is it's painful. I mean, it's not fun to do this. I mean, I actually thinking back to how we started this discussion, right? You asked me what I wanted to become, and I said like an astronaut or like a physicist. I also knew what I did not want to become, and that was to be anyone who has to do a lot of writing. I hated writing when I was in school. So like writing these essays in my German classes was torture for me. And well, look what we are doing now the whole day. But, yeah, writing grants, writing patents, writing papers. Yeah.
Do you find that there's certain days or certain times of day that you write better than others? And times when you find it really hard to actually put pen to paper or fingers to keyboards?
I would say what's hard, the problem is it's hard, the time is hard for me if the deadline is not pressing. So I tend to get this stuff done last minute, which is not something I recommend. But I guess it just reflects how much I don't enjoy the writing aspect for grants, etc.
Yeah.
So
thinking of the challenging of writing and not liking writing, what have you actually found the most challenging aspect so far in your career? Most difficult time maybe, or the most challenging aspect? Ooh, that's a long.
It's. I think it's actually. What's the right term? Like dealing with conflicts in the lab. That I find is one of the hardest things, right? You see both people's perspective, and you still need to kind of mediate between them. And yeah, I think that I find is kind of a hard.
Have you ever had to give someone a formal notice?
I fortunately never had to do that, but yeah, I would definitely. That's also something I would. Yeah, would be very hard for me.
Yeah, this is very personal, and generally, I think most our staff are very good staff and likable, but maybe not always best at their jobs, which can become a problem. Yeah, people in positions, two sort of different things. Okay, so going from the bleak that we had there, what about the most? What do you think in all your careers being the time that actually, if you could just live that moment time and time again? And you're like, what period of time would that be?
I mean, it's certainly, I think there are many of these short events, right? It's this, the minute you get, or the hour or the day you get the notice of a grant you got, or your paper got published, or like you get really the positive feedback of something finally working, like the experiment. The first time we saw like the pan-expansion microscopy images or something like that. That is these moments are just are just magic, right? And that's what keeps you going.
Be honest. Which is most exciting, seeing that first result on the microscope, or having it accepted as a publication?
I think it's the seeing it the first time on the microscope that's realizing that, it
has to be.
Yeah.
Because that's the first time it's really real, and you'll get it published.
Yeah. The realization that you have something really cool here, or that this is the, like, I mean, from a subjective perspective, that's the best image ever taken of this particular kind of sample or something like that. You know, that's a great feeling.
So, and you mentioned there, maybe seeing that big Pan-X image, thinking of big things. What's the next big thing in microscopy? What do we need to solve? What if there was no limitations, what would you enable?
I think one of the, I don't have an answer to that, but I think one thing which I really find one of the biggest challenges is this move towards quantitative biology, quantitative imaging, right? So this is obviously important. I think we all know that, but to actually make this work that is truly reliable, reproducible, robust, and quantitative, that you can really that you know what you have there, and you don't need to worry. There's maybe still another artifact I missed in here, any of these things. This I find is a big remaining frontier, and will remain probably frontier forever. But this is something where I'm sometimes, for like this, the machine learning field, for example, is something which worries me a little bit there, because I'm not really an expert there. I think it has great potential, and I think it's super important. But at the same time, I think it's also quite dangerous. You can create a lot of things which might not be real, and you're not realizing, or if you don't know what you're doing there using these algorithms, you can,
I would hope the scrutiny will always be there, and then put into practice to check and back check. But I understand what you're saying.
Yeah.
Some quick fire questions. Are you a Mac or PC person?
I'm a PC person.
Early bird or night owl?
Night owl.
What's your favorite food besides, assuming it's not creme
Chocolate.
Chocolate. You just damned chocolate earlier, and now you're saying chocolate's your favorite food. Chocolate
cake. You know why? Why do you need that crust underneath? If it would just be the filling.
Milk or dark chocolate?
More on the dark side, yeah.
Okay.
Yeah, dark side. Dark.
Tea or coffee?
Tea.
Wine or beer?
Beer.
Ale or lager?
Both.
Ooh.
Just not at the same time.
TV or book?
TV nowadays.
What's your favorite? Do you actually watch any trashy TV?
I'm actually not really watching TV. I'm not watching like Netflix or like Amazon Prime or so. So I don't really know what's currently on TV, because I don't like the waste of the advertisement breaks. But do I watch any trashy TV or trashy shows?
Yeah.
I don't know. I'm not even sure I know what would qualify as trashy.
I know, because that would reveal mine. I'm not going to reveal my vices. That'd be I can't do that. That'd be worse, I think, if I was to admit my own vices. I
watch a lot of science fiction, or not a lot, but if I have the chance, I watch kind of science fiction series from the Star Trek universe, or I watch.
So that ruins the Star Trek Star Wars question. So I presume it was Trekkie more.
Star Trek, definitely Star Trek.
Yeah. What's your favorite? Not a Star Trek, but of all time, of any genre. What's your favorite movie?
I don't know, but I can definitely tell you the movie I watched the most was Pulp Fiction. And when I was an exchange student in Glasgow, I think my friends and we went 6 times to the cinema to watch it.
6 times?
6 times, yeah.
With the same people?
Essentially, yeah.
Wow, I did Batman 3 times, I think. But I can't believe it. 6 times.
Yeah, over like 2 months or whatever, how long it played, yeah.
So how are your dance moves?
Oh, terrible.
Pulp Fiction.
I'm not a dancer, not at all.
You can't do the John Travolta moves. So
I wouldn't even dare to try. I would be far too embarrassed to even try.
That you just put it's a lovely concept that you're going to get my beer vouchers at the next conference, and I'm going to get the Pulp Fiction, that famous scene at Pulp Fiction, just to see your John Travolta moves.
It would require a lot of beer to get me to that.
I just love that concept of it. If you could live anywhere in the world, where would you move to?
I don't know. I think I'm pretty open, but I have this attraction to islands. I need to talk to some psychoanalyst at some point where that comes from, but this is really, islands fascinate me. whether it's a Greek island or whether it's the Azores, whether it's an island here along the coast of New England, doesn't matter. I think islands are just fantastic.
Okay, that's just a perfectly good idea. And if you could meet anyone in the world, who would you meet?
I don't think in these categories. I'm not somebody who I don't know. I'm not I don't believe so much in the genius of an individual. So I don't really put individuals on very high pedestals. So for me it's more my focus is much more on the team or like on a group having achieved something. I'm not idolizing very easily.
So coming back to work, because we're actually very close to the hour mark already.
Okay.
It's gone way too fast. Actually, I have to ask, just before we go back to work, you say you make desserts. I would say this is not a dessert. This is not a cake. I don't know. I think this is something you would do at Christmas, but surely it should be a
gingerbread house.
Okay, so it is gingerbread, but that's a microscope, not a house.
It's a gingerbread microscope, yeah. And the best part, it was operational. So we actually made lenses out of sugar here.
You made the lenses out of sugar?
Yes, you can do that. Yeah, so I forgot what the name is, but we essentially got this confectioners or like a sugar that you can look up recipes how you make hard candy, like the rock candy kind of thing, and it's clear, right? If you don't put any dye in it, and it's fairly clear, and it has a refractive index which is different from air, so you can form it. So then we essentially put that into a mold which was roughly a circle, and then we popped it out when it was hard, and you can polish it very easily because you just need to make it wet. So you just polish it between your hands and gets this nice smooth surface. I think I actually showed you an image here.
So I was just saying, I did not know what this image was, but now I do. So is this an image? So that was for those listening, there was a gingerbread house equivalent, but a gingerbread microscope.
Yeah, so that is an image taken with that microscope.
This a ruler?
That's a ruler underneath, yeah. So we imaged that ruler through that microscope lens, essentially. There's another image of the lens, actually, you can see.
So now, before we go to the image of the lens,
yeah,
you're a microscopist, you publish, you show your images. I would like to point out, you are showing me a ruler and no evidence of magnification whatsoever. There's no scale bar on this or nothing.
Oh, the ruler is yeah, actually, sorry, I didn't want to swamp you with images. I have an image of the ruler. You want to see the image of the ruler? Non-magnified? I have that.
Yeah,
here.
Go on.
Okay,
yeah, go for it. Just put it in your background. If you could put it in your Zoom background.
Okay, that just takes a minute to get that.
I would like to point out to the non-listener that this image has a few aberrations in it.
Just a few. I think it's nearly perfect.
And the lens, just while you're waiting, this is, I presume, the lens that you made, is it?
Yeah, that's the lens, yeah.
Good grief. And then just while George is looking for that, George also sent this picture, which suggests he actually shrunk himself and blew up the Nutella jar. So he's obviously used expansion microscopy to blow up a Nutella jar and hugging it.
Exactly, yeah. So that's the real motivation behind expansion microscopy, and have more desserts.
I can't wait for the time you blow a cell up so big or a bit of tissue that you can walk into the tissue. What is that, 10,000?
That is still our dream, like for a kind of a educational project, to expand the sample so much that you actually have the cell like that big or so, and you can actually just put it in a museum rather than we actually we got samples now. My student showed me samples that on the slide, on the petri dish, they're expanded so much you can just image them with your cell phone. it's amazing. You can see them just with your cell phone camera.
And enough fluorescence there to see that?
And not fluorescent, that they have some kind of scattering contrast in that particular case, and just it's amazing. Sorry, there.
Because the next thing, you talked about your love of making desserts.
See, here's the ruler.
Ah.
And it was a metric ruler, so this is a centimeter scale bar. I guess yeah. So yeah, the magnification is not that impressive with the microscope. I have to. Yeah, there's the number 5.
But it's a sugar-coated lens. Come on.
It's a solid sugar lens. Yes, you're right. Yeah. And it was our first attempt, so that was we need to repeat that experiment. The problem is after a few days, the microscope collapsed. So I think the humidity was too high.
So, George, we are over the hour. I have to say, thank you for joining me today. It's been entertaining, inspiring, and as inspiring as your work, actually. I can't wait to see the next developments that you come out with. I can't wait to see your next publications and you presenting them, because that's what really sells it and inspires as well. So George, thank you so much for joining me today.
Oh, thank you very much for inviting me. It was a pleasure to chat with you today.
That's a pleasure. And everyone who's listened or watched, do have a quick flick at the YouTube. If you don't have time, just go to those last few minutes to see the gingerbread microscope, which is surreal. But thanks for listening. Don't forget to listen to the others from Alison North, who's one of the early ones that we podcast with. And also don't forget to subscribe to whichever channel you're listening to. So George, again, thank you very much.
Thank you.
Thank you for listening to The Microscopists, a bite-sized bio podcast sponsored by Zeiss Microscopy. To view all audio and video recordings from this series, please visit bitesizebio.com/the-microscopists.